Excited at the progress that's been made in the past 20 years. It's really been a remarkable time in terms of biomedical research. 20 years ago, there was very little known about the innate immune response to viruses, how viruses and other pathogens are initially sensed, and how those pathogens then trigger an immune response. With the past 20 years, particularly the past decade, the concept of pattern recognition receptors, toll-like receptors, cytosolic sensors for RNA viruses have really been part of an exponential growth in terms of research. Although a lot has been known about sensing and response to RNA virus infection, really these initial steps in the triggering of the immune response, there's been much less that's been known about DNA viruses and how they are sensed and respond, and almost nothing that has been really known about how retroviruses are initially sensed in infected cells. And this area, of course, has changed dramatically and has grown tremendously over the past couple of years. And I'm going to talk today about the interface between interferon activation and the host response to a human retrovirus. And that human retrovirus is human HTLV-I, human T-cell leukemia virus, which, like its cousin, human immunodeficiency virus, uses a number of very ingenious strategies to maximize expression of its genome, including multiple splicing, ribosomal frame shifting, proteolytic cleavage of polyproteins, all aimed at maximizing the generation of not only structural but a number of regulatory proteins that control its early steps in replication and pathogenesis. HTLV-I, I think as most of you know, was the first of the human retroviruses identified by Bob Gallo and colleagues. It's estimated that up to 20 million people worldwide are infected with HTLV-I, and most of them don't know that they're infected. A small percentage of these asymptomatic carriers will go on to develop disease, and there are two main types of disorders that are associated with HTLV-I infection: the adult T-cell leukemia, which is being driven by one of the viral proteins called TAX. It results in a CD4, CD25, and often FOXP3 positive T-cell leukemia, shown here as a multinucleated flower cell. Also associated with HTLV-I is the disease tropical spastic paraparesis, an autoinflammatory disease and a myelopathy that can result in severe brain lesions and loss of motor skills in the periphery. This one is, of course, associated with a T-cell proliferation. This disease is associated with a high viral load and infection and continued inflammation in patients. So also like HIV, studies a couple of years ago showed that HTLV-I can infect dendritic cells, and as a consequence of infection of cells of the myeloid lineage can lead to transmission and transformation of CD4 positive cells by conveying the virus into these T cells. And there are a number of consequences associated with dysregulation of the myeloid cell compartment, including chronic infection associated with innate immune impairment, reduced interferon antiviral responses via SOX1, and upregulation of other negative regulators of the interferon response. This performed by Stephanie Ollier a number of years ago, and also an impairment in DC and macrophage differentiation and monocyte depletion. So we entered into this study with initially asking a very simple question, one that we didn't know the answer to. What's the nature of the innate antiviral response in cells infected with HTLV-I? And we used as initial model the primary monocytic cells. We set up a number of assays that could detect binding of HTLV-1, its early viral RNA entry into cells, various assays for the reverse transcription process, and then assays also to look for maturation of the virus and virion expression. The initial experiment was performed, our initial experiments were performed with a purified stock of virus derived from MT2 cells, and we could show that we can get increasing infectivity of these monocytic cells using this purified virus stock. Intracellularly, we could identify that RNA was getting into these cells, although over time the amount of viral RNA decreased. If I fall off the stage, please pick me up and bring me back on here. But I'm trying to see these slides here. And then as we use some of these assays, I'll anchor myself here somehow and see if I can stay here for a while. So initially we looked at 3 hours, 24 hours for various signs of virus replication. And in fact, we could identify binding of up to 90% of binding in 90% of the cells. If we used an anti-GP46 antibody, we could block all of that binding. We could identify, again, intracellularly very nice viral RNA in these populations and P19 expression, a matrix protein. But in fact, we could not find in these monocytic cells any evidence for TAX expression, no evidence for de novo viral protein expression, no evidence for protein virion production. That being said, we could identify very quickly evidence that there was a strong antiviral innate response being triggered in these cells. We could identify phosphorylation of IRF3, one of the initial transcription factors involved in the activation of interferon genes at 24 and 48 hours. We could also identify STAT phosphorylation, STAT upregulation, and upregulation of various interferon-stimulated genes involved in the initial antiviral response. As we looked further at these cells, we were quickly recognizing that these cells were undergoing apoptosis. They were undergoing cell death. We could identify that by staining for annexin 5 positivity, and using patient samples from 5 different patients, leukapheresed for these studies, we can show that there's a dramatic increase in the level of apoptosis in these cells that are being infected with HTLV-1. So abortive infection combined with triggering of apoptosis prompted us to look a little bit more closely. We could show that caspase 3 was being cleaved and activated. We could show that mitochondrial depolarization was occurring in these cells by diOC6 staining. And also in terms of some of the BCL2 family members, we could show that the pro-apoptotic protein BAX was being upregulated transcriptionally in these HTLV-1 infected cells. These phenomena prompted us to, or reminded us of the characteristics of HIV infected cells, and were reminiscent of the activity of SAM-HD1, a deoxynucleoside triphosphohydrolase activity that has been shown to be the latest, if you will, in a group of intrinsic host restriction factors that limits HIV replication. And SAM-HD1 joins the family with ApoBec, TRIM5A, and Tetherin as playing roles in blocking HIV replication. As probably many of you know, HIV-2 and SIV express a protein VPX which can nullify the activity of SAM-HD1, and SAM-HD1 can play a role in the limitation of HIV replication in resting T cells and dendritic cells. So SAM-HD1 protein contains a number of key motifs, the sterile alpha motif, an HD domain, and serves as an enzymatic activity that hydrolyzes the deoxynucleotide triphosphate pool which are going to be necessary for the early steps of reverse transcription. At high levels of SAM-HD1, relatively low levels of virus replication occur, whereas inactivation of SAM-HD1 can lead to high levels of virus multiplication in the HIV infected models. SAM-HD1 is also regulated by phosphorylation. Phosphorylation by CDK1 at S phase inactivates the activity, whereas when you get into G2M and G1 phase, the dephosphorylated form of SAM-HD1 will aggregate, and that will actually impact on the deoxynucleotide triphosphate pool. So, and it, like many other of the intrinsic factors, are upregulated by interferon regulation. So we set out both in cell line models and also in primary monocytes to investigate the role of SAM-HD1. As we knocked down SAM-HD1, we could find that we were also able to decrease the level of, not only the level of SAM-HD1, but also the level of apoptosis occurring in this HTLV-1 infected models. And also now adding back exogenous deoxynucleosides to overcome the effect of SAM-HD1, we could show that as we increase the amount of deoxynucleosides added into the culture system, that we could bring down the level of apoptosis in these HTLV-1 infected monocytic cells. In light of some other recent studies that I'll get to in a moment, we've also looked at the pyroptosis in these cells, recognized or defined by caspase-1 cleavage and cleavage of pro-IL-1 to active IL-1. And we could show that in the HTLV-1 model, there's evidence for caspase-1 cleavage, pro-interleukin-1 cleavage. That can be inhibited with the specific caspase-1 inhibitor, YVAD, but we did not see a dramatic decrease in the level of cell death, arguing that at least in this model, we're not seeing a strong component of pyroptosis in the cell killing that's being seen in the HTLV-1 infected cells. So this is another slide. This is the start of some studies that we conducted to show that most of the HTLV-1 DNA that is generated in these abortively infected cells remains in the cytoplasm, or the total fraction of the cells doesn't get into the nucleus. Whereas if you knock down SAM-HD1 activity, or you add back deoxynucleoside to overcome the influence of SAM-HD1, that you can actually get integrated proviral DNA in these infected cultures. So there's a relationship here between levels of SAM-HD1 and the progression of the reverse transcription and integration events that are occurring here. So we set up a number of primers to detect the initial strong stop DNA associated with reverse transcription, as well as other primers that would detect elongation of the minus strand of DNA being generated during reverse transcription. And we've also added in AZT in some experiments to block the elongation of DNA. And you can see here that in these infected cells, we can pick up very easily high levels of strong stop DNA corresponding to HTLV-1. But if we add in AZT to an HTLV-1 infection, that we are progressively decreasing the levels of elongating DNA, and we're also slightly impacting levels of apoptosis. So now if we do a knockdown of SAM-HD1 in these primary cells, we can see that, again, the process of reverse transcription seems to be progressing relatively normally, in that we can now identify not only strong stop DNA, but also the elongating DNA from the replicative intermediates. So to try and get an idea of, I may have missed a slide here. Okay. So to try and get an idea of what was actually being sensed in these HTLV-1 infected monocytes, we generated a probe that mimicked, it's a 90 nucleotide probe that mimics the a section of the strong stop DNA. It's biotin labeled, and then we used streptavidin beads to pull down to identify those cells that were expressing this strong stop DNA. We introduced this into cells. And either the single-stranded DNA or a double-stranded version of this 90 nucleotide fragment from the strong stop DNA could also lead to significant apoptosis in this cellular population. So the fact that it appears to be a reverse transcriptase intermediate DNA that is being sensed and causing apoptosis in these cells led us to look at the relationship between the apoptosis that we're seeing and the STING adapter. STING is an ER-resident sensor of intracellular foreign DNA and can complex with the kinase TBK1, leading to IRF3 phosphorylation and activation of the antiviral response. Of course, type 1 interferon then released leads to stimulation of the JAK-STAT pathway, which you've already heard a good deal about, and now upregulation of the triggering of this antiviral state. So to investigate a relationship with STING, we also did a knockdown here of STING and asked the question, does this also impact on apoptosis in the HTLV-1 infected cells? And as you can see, we significantly knock down the level of apoptosis in these cells, and there's a strong correlation here in the multiple patient samples that we've used between inhibition of STING and inhibition of HTLV-1 induced apoptosis. We also used our DNA, our biotin labeled DNA, to introduce that into cells and look at the association between STING and this strong stop DNA, and we could identify with either the single strand version of this DNA or a double strand version of it that we could actually get an association here between the DNA and STING. What we also noticed in this is that the single-strand DNA, but not a scrambled control DNA, is upregulating BAX, which is one of the characteristics that was seen in that first 3 hours or so of post-infection in this population. And as we went on in these studies, we then were able to show that HTLV-1 infection could lead to sort of P24 expression in these cells. We could get IRF3 phosphorylation and BAX upregulation, but if we knock down STING, we are significantly impairing those interactions, and we're also not affecting so much, as I say, the amount of IRF3, but the phosphorylation of IRF3. The fact that we're seeing an interaction between IRF3 and BAX was reminiscent of a study that was conducted 2 or 3 years ago by the group of Ganesh Sen, and he showed that in Sendai virus infected cells that an IRF3 complex was formed between IRF3 and BAX, and leading then to a novel form of apoptosis. So we set out here to examine whether in HTLV-1 infection or whether transfection of the strong stop DNA would actually lead to the generation of this IRF3 BAX complex. And in fact, if we do an immunoprecipitation of BAX and then probe for IRF3, we can find that association of IRF3 with BAX. The same thing occurs if we now have the single-stranded DNA transfected into cells. We can identify this BAX IRF3 complex being formed in these cells. Knocking down STING also now has the impact of decreasing the level of the formation of this BAX IRF3 complex, as seen here first by an immunoprecipitation with BAX and then coming back and blotting for IRF3. And likewise, as we see again, this decrease is impacting on BAX. So we wanted to complete the circle of this study and essentially also did a knockdown in these primary monocytes of the BAX pro-apoptotic protein. We could show that we are reducing the amount of apoptosis in these cells by knocking down BAX, and then by gating on CD14 positive cells and those cells that are expressing high levels of BAX protein, we were then able to show using an Exon-5 staining that virtually all of those cells that are expressing a high level of BAX are also those cells that are undergoing apoptosis. So the similarities here with HTLV-1 DNA have been repeated using a DNA fragment corresponding to the strong stop DNA of HIV. And the results here are very similar, that if we introduce now a single-stranded DNA from HIV, we also can identify a formation of a complex between IRF3 and the BAX protein, and a triggering of apoptosis in these monocytic cell fractions. So our conclusion here was that HTLV-1 infection induces apoptosis and a strong innate immune response within a couple of hours in this primary monocyte culture, and it's occurring in a SAM-HD1 dependent manner, because if we knock down SAM-HD1, we're able to eliminate aspects of the triggering of the antiviral response and also apoptosis. Silencing of SAM-HD1 by RNA interference inhibits this apoptosis, and that can be reversed by the addition of exogenous DNTPs that also reverse the effect of SAM-HD1. Introduction of a biotinylated 90 nucleotide reverse transcriptase intermediate from the 5' region of the HTLV-1 LTR is able to induce apoptosis. It does that by, in part, complexing with the DNA sensor STING, and this mediates activation of the host antiviral response via IRF3. This apoptosis is being driven, at least in part, by an IRF3 BAX complex that is being generated in these HTLV-1 infected monocytes. And finally, we conclude that these studies identify a link between SAM-HD1 host restriction factor, the sensing of retroviral transcription, not as an RNA, but as a DNA intermediate, and that this involves the adapter protein STING and the initiation of apoptosis driven by IRF3 and BAX. So this is a schematic of our conclusions at this point, and basically with sufficient deoxynucleotide triphosphate pool, incoming retroviral RNA will be converted into double-stranded viral DNA and integrated. As you saw, if we SAM-HD1 essentially poisons that DNTP pool, depletes that DNTP pool, resulting now in the accumulation of a reverse transcriptase intermediate DNA, usually mostly a small DNA corresponding to strong stop DNA, and that strong stop DNA can interface with the STING adapter, leading to IRF3 activation, triggering of interferon response and the whole interferon cascade. But at the same time, we're also seeing evidence for the generation of an IRF3 BAX complex, which was shown by Ganesh Sen, actually localizes to the mitochondria, contributing to depolarization and then activation of caspase-3 mediated apoptosis. So this was a very nice story. We're very happily going along with this study. And as you know, the last couple of months have seen a flurry of studies related to HIV and innate sensing of retroviruses by various sensors. One sensor identified by Soren Paludan in Denmark is another DNA sensor, IFI-16. Warner Green's group recently identified this in HIV-infected T lymphocytes, and a number of other groups, including, or particularly James Chen's group, has identified now a new cyclic GMP-AMP synthase that is playing a key role in STING-mediated apoptosis. So these papers have really sort of focused in on this early response to innate immune responses to viruses. And CGAS is a very interesting enzymatic activity. It generates a dicyclic GMP-AMP, generating a compound called GAMP, generated actually through a 2'5' linkage group, and is structurally quite similar to oligoadenylate synthase and its ability to generate a 2'5' oligoadenylate, which also plays a key role in stimulation of the interferon response. So it's thought now that the generation of GAMP is actually part of the mechanism of activation of STING. There have been a number of papers on the biology and the structure of STING and CGAS in the past year. And essentially, therefore, we went back and asked the question whether CGAS is playing a role in the triggering of the apoptotic response and the antiviral response. If we knock down CGAS in these primary monocytes that are HTLV-1 infected, we get a nice knockdown, and again, we are able to significantly knock down the level of apoptosis that's being driven in these particular cells. What's also been quite interesting is we've used our pull-down technology using the biotinylated DNA fragment to look at the possibility that we might be identifying or recognizing other sensors. And you can see here that we can find association with STING, CGAS, IFI-16, and to a limited extent MRE11 in cells that are HTLV-1 infected and now expressing and now associating with, in fact, this DNA fragment. So the fact, however, and I don't have the slide to show that, is that there is little evidence for direct binding to STING using recombinant STING proteins. So we think we may actually be looking at a complex. Our original model may have to be modified. So we're actually not only looking at these retroviral DNA intermediates interacting with CGAS and STING, but also potentially interfacing with other DNA sensors, perhaps in a cell-specific manner, then leading to the triggering of the antiviral response through STING and IRF3, and potentially as well, and this is an area that we're investigating right now, is the relationship between the sensing of retroviral DNA intermediates and the triggering of aspects of the inflammatory response. So I'll just conclude and say I wasn't too jealous of these other great papers that came out in the last couple of months, because we've also had a couple of papers that have come out on this subject, most of which I've just covered for you, and they both have great looking covers, which is always a nice bonus for this particular work. So I just want to conclude by thanking a number of people in the lab, particularly Julian van Grevenenge, who actually was at VGTI Florida, but his wife is French-Canadian, so he actually moved back to the lab in Montreal because he wanted to stay married. And he's actually was a real driving force in this project with HTLV-1. Alex Says is a graduate student in the lab working on this project. David Olenier and Cindy Chang in VGTI Florida have been progressing with this project, and a number of other collaborators who have contributed to this. So with that, I'll thank you very much, ladies and gentlemen, for your attention.
Thank you, John. Any question from the audience?
The replication of the virus with production of Tax protein, and I mean, if you reduce the apoptosis, how does this thing impact on the outcome of the disease? I mean, and because my question is SAM-HTV1 can be considered a target, but which way to go? I mean, inhibiting or promoting its function? That's something I really didn't understand.
Yeah. So the question of SAM-HD1 involvement and virus replication. If we knock down SAM-HD1 in that monocytic cell culture, we do integrate some level of integrated proviral DNA using ALU-PCR. We're doing experiments now with monocyte differentiation in dendritic cells differentiated in vitro. Those cells have a level of also replication that occurs, tax expression, integration of proviral DNA, and they also are displaying just a decrease in the level of SAM-HD1. SAM-HD1 as a target, yes, it certainly is one of the targets that could be, I would say probably you would want to increase the level of SAM-HD1, block virus replication. On the other hand, you may impact cell cycle because it's intimately linked with cell cycle control.
Thank you.
C'era Martina e poi Maria.
Yes, thank you. Very nice talk. I was also in light of the data, literature data that Sandra Pellegrini mentioned before. I was wondering if there is any evidence on dysregulation of SAM-HD1 function in the context of the human endogenous retrovirus infection and chronic activation of immune responses leading to hyperimmunity.
Yeah. Well, certainly, as Sandra mentioned earlier, SAM-HD1 is one of those nuclease activities that, if inappropriate, if mutated, can contribute to development of autoimmunity, particularly Aicardi-Goutieres syndrome, and so on. As yet, I think I don't think as yet there is any evidence that SAM-HD1 plays a role in regulation of the endogenous human retroviruses, but that's certainly a very good question to pursue.
Very short.
Very short question.
Okay.
This is curiosity. Is the addition of CD4 cell to the monocytes able to rescue the infection and possibly can this be envisaged as an escape mechanism from the
I see. Yes. Some of these experiments have been done by co-culture with CD4 positive cells, and that actually is an area that we are looking at to get an idea of what is the difference between infection of a monocytic culture versus a T cell culture, given particularly that based on the Warder Green studies that we're seeing an apoptotic predominant phenomena for depletion, whereas he sees a pyroptosis predominant phenomena for loss of CD4 positive T cells. So those are ongoing things. So.